Yes. Okay. Hi, welcome everybody. I'm Serdar Bulun. I'm the chair of OBGYN here at Northwestern University. And I just would like to let everybody know that right now we have over 500 attendees virtually joined us. And we are among a lot of family and friends. This is a great occasion. And I'm really excited that Francesca won this award. And I never met Roger Short personally, but he was well known in the field of reproduction. My late mentor, Paul McDonald, was good friends with him. And from the stories Paul told me, he was a very brilliant, creative, and a colorful scientist. So I would like to introduce Dr. Melissa Mann, who's the current vice president and incoming president of the Society for the Study of Reproduction from University of Pittsburgh. Melissa, thank you so much for being here.
Thank you. It's great to be in Chicago, and it's great to have so many colleagues online. So the Roger Short Medal and Lectureship was established by the Society of Reproductive Biology, the Society of Reproduction and Fertility, and the Society of the Study of Reproduction, together with Doctors Marilyn Renfree and Jock Finley. Now, Roger Short was said to be a very inspirational reproductive biologist. He was described as a monumental 19th century thinker and a great man of science. And this is in part due to his depth and breadth of research in reproductive biology. And he was hugely supportive of young investigators. So with this interest, together to help put reproduction on the world map, we came together with this award. How do I forward? Sorry. Oh, okay. So in a little write-up about Roger Short, Doctors Cooper and Hendelson said that Roger Short's focus on reproduction had the largest themes across the widest content. And I want to tell you three little anecdotes about Roger Short and his science. So he, when he was a graduate student, went out to find his experimental model, which started out with the badger. So he had to go with an old badger digger, and find his samples to do for research. And it happened to be that they had a sow. And when he was looking at the reproductive biology of the tract, he discovered that inside the uterine tract was the diapause badger blastocysts. And he claimed they were the pearls of life, and also declared at that point in time that he was going to be a reproductive biologist. And he did not limit his studies to one species. So here I've listed a number taken from his record of 400 publications, the different topics, including from the spotted hyena, the elephant, the wallaby, humans, birds, sheep, equids, camels, great apes, the red deer, the red fox, and the human population. In one interview, he was asked, well, how do you go about studying elephants? And he said, well, it was early days. So he got his crossbow and he manufactured darts that contained a chemical that would anesthetize the elephant. So he shot at these elephants and then recovered the tissues that he needed for his science. And he, at that point in time, was near the equator and said that he walked across the equator more times than most people. There's one other story that I want to tell you about that I think is quite telling and endearing for Dr. Short. When he was a graduate student at first going to university, his father sent him a letter with a quote by Emerson. And it said, paraphrasing, explore the truth and beauty. Be bold, be firm, be true. Be fast to your intellect. Explore, explore. And he lived that mandate. But what's so touching is he kept that letter in his pocket for his entire life. So with this amazing scientist and the group we had together, we developed this Roger Short Medal and Lectureship. It is a prestigious international award that can be celebrated around the world to recognize early career researchers who possess the same spirit of discovery, creativity, and imagination as Roger Short, and who have made outstanding discoveries of the field of reproduction. The third competition will be coming up soon, so please keep your eye out for this. The second award winner of the medal is Dr. Francesca Duncan. She did her PhD at the University of Pennsylvania with Carmen Williams. She then went on to do a postdoc at the University of Pennsylvania with Richard Schultz. For a second postdoc, she came to the lab of Teresa Woodruff, where she made many seminal discoveries. To start her independent career, she started at the University of Kansas Medical School before she was recruited back to Northwestern. She's an associate professor in obstetrics and gynecology. She's the Thomas J. Watt Memorial Professor in Reproductive Science, and she's the co-director for the Center of Reproductive Sciences at the Feinberg School of Medicine at Northwestern University. Please welcome me and celebrating your award. Congratulations.
Well, thank you so much, Dr. Bullen, for kicking us off. And thank you, Dr. Mann, for that beautiful introduction and insight into Roger Short and the spirit of this award. It is truly an honor and privilege to be able to be this awardee and present our work and to follow in this footsteps of explore, explore, explore. And so today I'll be telling you really, my lab is 10 years old, so I made it just in the nick of time of the eligibility criteria of this award. So it allowed me an opportunity to really reflect back on this past decade and really the work that we've done on ovarian aging from basic mechanism to clinical translation. But before I start, I want to really do a few thank yous. And so it is with tremendous gratitude again for the 3 societies that have recognized Dr. Short and his contribution to our field, but also to the legacy of those that follow. I also want to acknowledge Bruce Murphy, who is the person who nominated me. And so I will say that while I've never worked directly with Bruce, I have always admired his work as someone who studies embryo development, but also ovarian biology. And so it was really humbling when he emailed me to ask if he could nominate me for this prestigious award. And of course, Bruce was joined by reference writers, and I'm forever grateful to Dr. Teresa Woodruff, who is a longtime mentor and now a colleague of mine, as well as Dr. Raj Kumar, who is a mentor but also collaborator of mine. So thank you so much to them. So I also am tremendously grateful for our entire field. And I realized as I put this together, this probably wasn't the best use of my time. And that also I can't fit everyone who has really impacted me throughout my career on one slide. I had the library look at my publications, and I've collaborated with 700 different people in my career. So there's no way that's fitting on one PowerPoint slide. So this is an aliquot of our field. And what I've learned in putting this together and really reflecting on my career is that it really takes a village to build, nurture, and grow a career. So I thank all of you who have been involved in these various capacities for my career and helping support me. I also want to thank my local community. So a big theme that you'll hear in my talk today is that the nest matters. And so that nest of this local community where I do my work has been really instrumental to my career. So I'm extremely grateful for the Department of Obstetrics and Gynecology with a great group of colleagues in the Division of Reproductive Science and Medicine. But we also house really important programs like the Center for Reproductive Science and the wonderful leadership that supports our center. So I thank all of them. And then the terrific Northwestern Master's program. Many of those individuals who are part of that program who have contributed to the work that I'll talk about today. And I'm also really enriched with partnership with the Longevity Institute that we have at Northwestern, which really couples my interest in reproductive health and women's health together with human longevity. And then academia is also really exciting and the flexibility that it affords you. And I have 10% of my effort that is with the Buck Institute in California, which is a private research institute focused on aging. And I'm really grateful to that community for welcoming a reproductive scientist to really broaden the discussions of the work that we do. Now, the story started in 2014 with a pristine empty lab at the University of Kansas Medical Center. And it is with a lot of humility and gratefulness that I thank my partners in discovery. If it wasn't for them, there would be no talk today. And so I'm really grateful to this tremendous group, and it's been really a privilege to watch generations of a lab evolve and to see all of these brilliant, enthusiastic, genuine people lead really successful careers of their own. So this field has a bright future because of all of you in it, and I believe you are all simply amazing, so thank you. And then this is tremendous gratitude to my loudest cheerleaders. So when my dad, when he was alive, was definitely one of those. I want to thank my husband and daughter for their continuous support, as well as my mom. And I'm an only child, but I'm very lucky to have my biological mom and my scientific mom, both who are in the center of the audience today. So thank you for being my loudest cheerleaders. Okay, so now into the science. So what I hope to cover today in our time together is really an overview of the topic that I am very passionate about, which is female reproductive aging. And I'll tell you about our foray in really opening up the discovery of the aging ovarian microenvironment. And I want to share with you actually some new work on why does this happen, but what are the biological consequences? And this has really inspired me as I look towards the next 10 years to think about how we can translate what we've found into the clinic. So how can we modulate and measure the ovarian microenvironment? And I'll end with some of new frontiers that we've developed to be able to model this critical part of the ovary. And I want to point out, this is actually the first paper I published as an independent investigator that shows ovarian fibrosis. It made the cover of Reproduction, which I was very grateful for, but really changed the trajectory of my research program. So very proud of that work. So for those of you who know my research, you probably see this graph every single time I talk, but this is really what drew me to the field, which is that the female reproductive system is really one of the first to show overt signs of aging in the human body. So what we know, as you can see in the blue line, is that across organ systems, there's deterioration in tissue function as we age. But the female reproductive system is unique in that it does this much earlier and more drastically relative to the other organ systems. So female fertility will begin to decline in women in their mid-30s, and reproductive function will cease completely at the time of menopause at around age 50. Now, in the ovary, this reproductive aging is characterized by really two main events. First, women are born with a fixed number of follicles in the ovary, and there's a natural winnowing of that ovarian reserve with age, and you can see that drop in follicle number in the red line. And you can also see that in these histological images. At the top, that's ovary from a 7-year-old. All of those circular structures are the primordial follicles. And then in the 29-year-old ovary, you can see there's that one follicle present. So even in young individuals, there's a drastic change in the number of follicles in the ovary. And this is accompanied by a corresponding loss in the quality of the eggs within those follicles. And you can see that in the blue line as an increased fraction of poor quality oocytes. And so again, for those of you who've heard me speak, you've heard this quote too, which is that the ovaries are probably one of the only organs whose peak potential is reached long before their first use. And so this is one of the clinicians in our field, Dr. Genevieve Neal Perry, said this, and it's something that has always stuck with me, a really powerful quote. So reproductive aging has significant clinical ramifications. So we know with age, this loss of egg quantity and quality can lead to infertility and spontaneous abortion. But for those women who are able to conceive, advanced reproductive age is also associated with dizygotic twinning, obstetrical complications, and birth defects. And this matters because globally we're delaying childbearing. This is data from the United States, but is really mirrored across the globe, where that the age at first birth is increasing significantly. So you can see that in the black and blue lines. But it's important to realize that it's also beyond fertility. We know that follicles, as they grow, they produce really important hormones like estrogen and progesterone, which are important for regulating downstream organ systems. So with age, we know that there's a decline in these hormones, and this can have adverse general health outcomes with respect to, for example, cognitive health, cardiovascular health, immune health, sexual health, and so forth. And so it's important to realize that while menopause is not a disease, we're really changing the landscape by which we're living. So what you can see on that graph is that the age of menopause has stayed constant for the past 200 years, or if not longer, at around age 50. But due to medical and health advances, our life expectancy has increased significantly. So in the US, most women will live to be about 80, which means they're going to be living about 3 decades in a postmenopausal state, experiencing negative consequences of reproductive aging. And so there's really, I think, an urgent need with significant clinical and health ramifications to really understand what are the mechanisms that underlie reproductive aging. And I like to think about that it is, I always say it's an exciting time to be in reproductive aging research, so I guess that's a good thing. But I think about what it was like 10 years ago when I was doing my postdoc in Teresa Woodruff's lab. And I attended the biology of aging course. And so that's me with that white skirt there. And what you can see is I was a lone reproductive scientist amid a really bright group of aging researchers. And I remember when we had to go around the room and explain what we did, there was a lot of skepticism about whether ovarian aging was actually aging when it was happening in really young individuals. And I looked to see what's happened in the past 10 years, and it's really a different time. And where we've built a community of practice. And so what you can see there is that now the Buck Institute, again, a private research institute, has a dedicated program on the Center for Aging and Healthy Aging in Women. We have a dedicated reproductive biology hub. Last year, there was a 2023 Biology of Aging course, and we were there representing reproductive science and teaching that to the aging community. At our societies, we're really good at having reproductive aging as a session at conferences, but now all of these aging societies also recognize this as an important area. And I've been really excited to be involved in launching a formal reproductive aging conference. We've also seen shifts in funding across the board where reproductive transitions are considered a key priority, and it is my true hope that this will continue. And we also have an amazing amount of awareness. So I am contacted by sort of the media very often, and I'm really glad to see increased awareness about this topic really throughout the media. And we also had a recent focus issue in Nature Aging, specifically on reproductive aging. So it is an exciting and dynamic time. I also just want to highlight that there are exciting events in this space in 2025. So I've co-organized an aging mechanisms impacting women's health through the NIA. Of course, we're still hoping that this goes through. It's scheduled for March. So please stay tuned for that, but that is available to the public online. And then we also have our 4th annual reproductive aging conference in San Jose. Okay, so back to the research. So I am an egg biologist by training and interested in reproductive aging. And when I started my lab, I was, again, thinking that I would pursue really understanding what is happening at the egg with reproductive age. And the reason for this focus, not only do I think the egg is the most important cell, but there's data from assisted reproductive technologies that really convincingly demonstrates that the egg is really at the root of age-related infertility. And so in this graph, this shows the percentage of fresh embryo transfers that result in live births in 2 cohorts of women across age. So in the solid line, in the dashed line, these are women who've used their own eggs to conceive. And in the solid line, these are donor eggs from donor women who are typically young, healthy women in their early 20s. And so what you can see is that if a woman uses her own eggs to conceive, the chance of bringing home a live offspring approaches nearly 0% in women in their mid-40s. However, if women use donor eggs to conceive, this maternal age effect is essentially abrogated. And so what this tells us is that the biological age of the egg is critical in dictating reproductive outcomes. And so when I started my lab, my goal was to understand, again, what are these changes that are happening in the egg? And a lot of work had looked at eggs that were either fully grown in antral follicles or those that had ovulated. But I wanted to look back in the development of the egg when it was starting to grow in the follicle to see, can we see some origins of changes that are already impacting the quality of that egg? And so we were interested, I wanted to take an unbiased look. And so we wanted to isolate follicles and perform RNA-seq. And again, I was interested in early stage follicles that are highlighted in those red boxes. And so what you can see on this images, on the left is a ovary from a reproductively young mouse. And on the right is a reproductively old mouse, which would be equivalent to a woman in her mid-30s, early 40s. And so what you can see off the bat is that there are fewer follicles in the old mouse, and that really phenocopies what we see in human. But there are still follicles present, and we wanted to go after, again, those early growing follicles that are highlighted in that red box. And the way we do that is we use insulin needles where we poke the ovary and we get those small follicles out of the ovary. And the people in my lab would often say it's really hard to get those follicles out of the old ovaries compared to the young ovaries. And if you look at the histology, you can see that that environment, or everything but the follicle, looks really different in the old ovary compared to the young ovary. So keep that in mind. We'll come back to that. Nevertheless, my lab always persists. And they were able to isolate these early growing follicles from young mice on the left and old mice on the right. And each row is follicles from an individual mouse. And so you can see that these early growing follicles look morphologically indistinct, although the environment, remember, looked quite different. So we did individual follicle RNA-seq, which I will say at that time was a huge feat. And what we were able to see is that based on our differential gene expression, there are about 2,000 differentially expressed genes. And based on this PCA plot, you can see that the old follicles segregated from the young follicles. So the old follicles are shown in purple and blue, the young follicles in red, orange, yellow, and green. So obviously we were interested in what was driving this change in gene expression that was associated with age. And through in silico analysis, we were able to determine that the primary changes were actually due to changes in the granulosa cells that were shifting the gene expression profile to a highly inflammatory or immune-like signature. And so all of these genes are not important just to say that they are all genes related to immune or inflammatory landscape. And on the left, where it's blue, that's young, and on the right, it's old. And so red is high levels of gene expression, and you can see that these follicles assume an inflammatory pattern of gene expression. So this observation, combined with the fact that it was actually harder to get these follicles out of the old ovaries, started us thinking actually very far away from the egg and started to think about this concept of inflammation and fibrosis. So we know that across aging, tissues, it's associated with chronic, progressive, non-resolving pro-inflammatory state called inflammation that happens in the absence of overt infection. And inflammation and aging are associated with multi-organ fibrosis. So if you think about this like cardiac, pulmonary fibrosis, which is really due to an excess deposition of extracellular matrix, in most cases, collagen deposition. And inflammation and fibrosis can be good, but if they are not properly resolved, that can lead to tissue damage and homeostatic dysregulation. So what started out as a side project was simply asking the question, do inflammation and fibrosis occur in the aging ovary? And again, this is not atypical of an aging tissue, but the ovary is doing this much earlier relative to other organ systems. And I think, again, coming back to that nest perspective, this work really shifted the perspective in the field where a lot of work had been done focusing on the egg, which again is a super important cell. But this changes the perspective that it's not just the quality of the egg, but the nester environment where it develops. So at the end of this talk, if this is the only slide you remember, we're good, which is that with age, we hypothesized that this environment of the egg was changing and that this would have indirect effects on egg quality. So the first thing we asked was whether or not the aging ovary is in fact inflammatory. And so these were some of our initial findings where we did qPCR in young and old ovaries and found that the old ovaries have a higher expression of pro-inflammatory molecules that you can see there. But not only do they produce them at the transcript level, but they're actually secreting large amounts of these pro-inflammatory molecules that we can actually assay in the conditioned media of an old ovary. So you can see there are very high levels of IL-6 being produced by this structure or this organ. We also made an observation, this was initially an observation made by an undergraduate in the lab, that there was a really unique population of cells that were highly penetrant in old ovaries. So every old ovary, we would see them, and we would never see them in young ovaries. And the way we identified these is actually they stain brown in a normally stained H&E tissue section. So they were extremely visible. And it turns out, and I'll come back to these because I think they're actually really important in the aging process, that these are multinucleated macrophage giant cells, which are typically found in other tissues in settings of chronic inflammation. So you can see here quantified in mice that these increase significantly with age, and we've also observed these in other tissues like non-human primate. So I'll come back to this because, again, this is a new area that we're really trying to understand what are these cells and their consequence in ovarian aging. So fibrosis typically follows an inflammatory state, and we knew that these ovaries seemed tougher because it was harder to get those follicles out. And so we did a simple stain, a histological stain, which detects collagen 1 and 3. And what we found across an aging series of mouse ovaries, as you can see in that red stain, is that there are fibrotic foci which really begin to expand throughout the ovarian stroma, such that the ovary at the oldest age is about 35% of that tissue is fibrotic. And we can confirm this histology using a biochemical assay called a hydroxyproline assay, which allows us to measure the amount of collagen in the tissue. And you can see that just like the increase in picrocereus red staining, there's also an increase in hydroxyproline, again, indicative of higher collagen. So we knew that this ovary seemed harder. We know that there's more collagen, but you can't really convince someone that something's just harder, just trust me. So we wanted to actually assay the biomechanical properties of the ovary. And so to do this, we performed a method called instrumental indentation, which is a mechanism where you can remove an ovary or organ from the body and use a probe to measure the amount of force required to indent that tissue a given distance. And so if a tissue is stiffer, it's going to take more force to indent that organ. And in fact, when we did this with young and old ovaries, you can see that it takes more force to indent an old ovary relative to a young ovary. And this was highly correlated to the fact that our old ovaries have more collagen relative to the young ovaries. Now, we've further gone on to see that it's not just collagen that's causing the changes in those biomechanical properties. In work that I won't be able to share today, we've done a tremendous amount of work on hyaluronan, which is a glycosaminoglycan, and we know that there are ages with that, or age-related changes in that macromolecule. But we also have done unbiased proteomics, comparing the proteomic signature of young and old ovaries. And what you can see here is that the young and old ovaries segregate by differential protein expression. And when we look at what are causing those age-related changes, there are many proteins involved in inflammation and fibrosis, again, echoing what we were able to show earlier, which with more focused technology. And many of the proteins that are changing in the ovary with age have been implicated in fibrosis in other organs. So I think the ovary is a really useful model to study fibrosis in other organs as well. So obviously this is all well and good, but all of this work that I told you about has been done in the mouse, and we wanted to always make sure that what we're doing is relevant to what happens in the human. And so obviously it's harder to work with human tissue, but what you can see on the left, this is follicular fluid profiles of several fibroinflammatory molecules, and you can see that these molecules increase significantly with age, and they're independent of other factors such as BMI. We've also looked at a tissue microarray of over 60 human ovaries ranging in age from half a year to about 60 years of age. These are just 3 representative cortical regions, and you can see stained with picrocereus red to detect collagen and fibrosis, and you can see that there's an age-dependent increase in the human ovary as well. So I've been giving this talk, and actually this is to the Aging research community, and this has really sort of been the #1 question that I've been getting recently. So I thought I would actually put this out here and start sharing some of my thinking in this space. So the question is, why does the ovary age earlier relative to other organ systems? And I think it's multifactorial. And, I'm not even going to consider the oocyte, but I think that's one contributor in that you have an organ that has an extremely long-lived cell. But if I think about the ovary relative to other organ systems, is that this is a highly dynamic organ that has a major challenge of managing cellular debris clearance just to achieve a normal, healthy function. And so what I want to just bring everyone onto the same page is that this is a very simplified diagram of what's happening in the ovary, but you have select follicles that are activated to grow, and only one in a monoovulatory species or a select few in a polyovulatory species will actually be recruited to grow and ultimately ovulate. You also have ovulation, which is the release of an egg from the follicle or from the ovary, and this requires a rupture event and a wound healing event. And then you have the rest of the follicle after it's ruptured that actually involutes and forms the corpus luteum that produces progesterone to support a pregnancy. But if there is no pregnancy, that corpus luteum regresses. So here's a challenge. So females, I told you, are born with a fixed number of follicles. There are millions of follicles, but only about a few hundred are going to ovulate in a woman's lifetime. So what happens to the rest of those follicles? Well, a lot of them are going to actually die themselves, or they'll activate and not make it to a final stage, and so they die too. So if you look at a normal healthy ovary, there's a tremendous amount of cell death that is taking place that has to be cleared. You also, as I mentioned, so even though you only have hundreds that ovulate, you do have hundreds that ovulate. So that's hundreds of times that this organ is going to wound and repair, and that's going to lead to scar tissue. And finally, you have this large structure, this corpus luteum, that has to regress. And in textbooks, somehow they just go away. But in reality, it's not so simple. And so you really have this organ that has to manage this amount of debris over a long period of time. And so we're really starting to think that these debris clearance mechanisms are what are starting to fail with age. And I won't have time to talk about it today, but we have interrogated point #2, and we know that wound healing and repair during ovulation is in fact compromised with age. But what I want to share with you is some new work that has been led by Dr. Aubrey Converse and her subgroup in my lab, where she's really looking at this point of corpus luteum as failed clearance of this structure as potentially a driver of aging. And so a shout out to the master's program. This is work that Aubrey's master's student has done this year, and it's really incredible work. So I want to kind of come back to this concept of failed clearance of a transient structure. So, and again, this started with observations, just like we found with robust observations of the presence of these multinucleated giant cells in the ovary, which is that there was a presence of a structure that seemed to be only present in old ovaries. So what I'll show you is that are labeled in these images as CLs. These are corpus luteum, remnants of ovulation. They're very large structures. I've already told you about the multinucleated giant cells that are shown in brown. These are these unique immune cell populations. But we also have that asterisk, and this is a structure that we've called a foamy corpus luteum because it has a very foamy appearance relative to the normal corpora lutea. And so if we count the number of these foamy corpora lutea, we see that they're, again, highly penetrant in the old ovary, and they're rarely found in the young ovary. And so this made us start to think that perhaps these are actually failed clearance of a CL. So they're a persistent CL in our old mice. And so to get a sense of this, to confirm this, Maddie and Aubrey's group did a labeling experiment where she super ovulated mice, and these are old mice. And then after the super ovulation, right after ovulation, she did a series of BRDU injections to label proliferating cells. And then we know that in this case, the corpus luteum at this time point would be proliferative. She then allowed these mice to age for 3 weeks and then harvested their ovaries and looked at what structures had these cells that were initially proliferating at the time of ovulation. And in fact, what she showed is that this foamy CL, this structure, actually was something that had persisted for 3 weeks. So it was an old corpus luteum, and you can see that because those brown stained cells are labeled with BRDU. And these, again, are highly visible in old ovaries, but not in young ovaries. So this is telling us that these corpus luteum are persisting for quite a bit of time. So we then started to think, could there be a relationship between the foamy CLs and ultimately the multinucleated giant cells? And so she did a really elegant experiment where she performed laser capture microdissection, which allowed her to look at these very precise structures in the organ at a transcript, ultimately at a transcriptomic level. So she was able to isolate corpus luteum, foamy CLs, as well as these macrophage multinucleated giant cells. She then, after isolating these, performed bulk RNA sequencing, and the results were really exciting. So what you can see in these principal components analysis is that our foamy CLs cluster transcriptomically very close to our multinucleated giant cells, and these are both far away from our corpus luteum. And what you can see here is that our foamy CL looks very similar transcriptomically to the multinucleated giant cells in that they are not steroidogenic like our corpus luteum, but instead they take on a more immune-like profile. And so this tells us that these foamy CLs may be, or persistent CLs, may be an intermediate between the CL and the multinucleated giant cells. Now, whether they are themselves macrophages or whether granulosa cells are taking on these luteinized cells, taking on immune-like signatures, we're still trying to sort out. But clearly this seems to be an intermediate before you get to those multinucleated giant cells. Now, Aubrey has done really tremendous work to study these multinucleated giant cells. This is a beautiful tissue-cleared ovary from an old mouse, and you can see that these multinucleated giant cells are really throughout that ovary. And due to time, I won't be able to highlight this work, but it is on bioRxiv. It's a really elegant paper where she also looked at laser capture microdissection to really understand the transcriptomic signature of this particular population. And what she was able to show is that this population is really shifting its gene expression signature to degradation, high energy production, and really unique immune processes as they're associated tightly with T cells. And so again, this is really consistent with the fact that this cell population may be dealing with this excess debris from persistent CLs. But what's also super exciting is that Aubrey has figured out ways to isolate these population of multinucleated giant cells. You can see them here in an isolated clump. We can also tell them because they're autofluorescent. And she can actually take what they're secreting and use that conditioned media to interrogate how they impact follicles and how they impact the stroma. And so she's done that where she's co-cultured follicles and somatic organoids separately with conditioned media from these MNGCs, and then performed RNA sequencing. And the details here are not so important, but what I want to highlight here is that in the follicle, these MNGCs are converting the follicle into an immune signature and the ovarian stroma into gene expression patterns for extracellular matrix remodeling. So this tells us that this population can actually influence inflammation and potentially fibrosis, again, coming in full circle of how this environment may be created. So this is really exciting work and is the area that our lab is focusing on now. But I think ultimately reproductive aging in the ovary is probably largely due to this fact of large debris clearance mechanisms. So back to kind of the story, we know that the ovary is inflammatory and stiff. The question is, who cares? And so I will give you 3 reasons why I think we should care. And this is work from Teresa Woodruff's lab many years ago, where she developed a really elegant system to be able to isolate follicles, encapsulate them in alginate. And the nice thing about this is that you can actually change the stiffness of the gel based on the concentration of alginate. And so a stiffer hydrogel would mimic an older ovary, less stiff hydrogel, a less percentage alginate would mimic a young ovary. And so what you can see here, here the range is 1.5% alginate, so stiff, 0.25% soft environment. What you can see is culturing follicles in a stiff environment leads to poor growth of the follicles. They're less functional, so they produce less estradiol. And importantly, that the eggs that they produce are not as developmentally competent. So follicles that grow in a stiff environment, only 5% of them make eggs that reach the 2-cell stage, whereas in a softer hydrogel, that amount is increased significantly. So we think that that aging ovarian environment is going to negatively impact follicle growth, function, and gamete quality. And this work has been followed up by Dr. Farnaz Amargant, who is a postdoc in my lab, and she actually determined that an old ovary is about 2% alginate, and she's done a really nice study to actually drive down into the molecular mechanisms of how the follicle is responding to this stiffer environment. So it's really nice to see this work pass through multiple generations. We also have shown that with age, you would expect if there's a fibrotic, stiff environment, that it's going to be hard for ovulation to take place. And in fact, what we see when we hyperstimulate young and old mice, we see that there's a greater amount of failed ovulatory events with age. And so here we're quantifying the amount of eggs that are trapped within unruptured luteinized follicles, as well as eggs that have clearly expanded in cumulus oocyte complexes, but fail to rupture in the time course of the ovulation that we were looking at. And so this you can see quantified here. There's more failed ovulations with age. And we and others, like Becky Robker's group, has shown that if you actually modulate this fibrosis, you can rescue this age-associated defect in ovulation. And so finally, I think the other example that's really important is that this environment may actually be permissive to ovarian cancer pathogenesis. So we know that ovarian cancer is most frequently diagnosed in postmenopausal women. And if you look at a 63-year-old ovary stained with our collagen dye, you can see that most of that tissue is fibrotic. And really elegant work from Joanna Burdett's group has shown that oviductal epithelium cells, where ovarian cancer originates, tend to prefer colonizing on 3D-rich collagen matrix, of which the aging ovary is one of those. And then in some initial studies, we've shown that ovarian cancer cells that are tagged with fluorescent proteins, RFP here, prefer to adhere to old ovaries compared to young ovaries. So while we don't think, obviously not all postmenopausal women get ovarian cancer, it could be a second hit or, again, a permissive niche for this pathogenic scenario. So because of all of these three reasons, we're really motivated to see if we can actually modulate these parameters in a therapeutic fashion. And this concept really came to light when we first showed that we can actually modulate stiffness by modulating the collagen content. So this is an ex vivo assay that I showed you before with instrumental indentation. We have our young and old ovaries that I showed you before, but we now added a third group where we introduced collagenase treatment to reduce the collagen content. And you can see that when we look at the Young's modulus or the stiffness, our collagenase treated old ovaries now look much more similar to a young ovary. So we can modulate collagen and we can modulate stiffness. So again, Dr. Amargant, when she was in the lab as a postdoc, wanted to do a proof of concept study of is there a therapeutic benefit to modulating ovarian stiffness. And so her premise was that if we took mice at around 7 months of age when fibrosis starts, and we treated them with a low-dose antifibrotic agent, that we would actually maintain a homeostatic environment and prevent high levels of fibrosis from happening. And so to test this, she used pirfenidone, which is a broad-spectrum FDA-approved antifibrotic drug that's currently used in the setting of idiopathic pulmonary fibrosis. And we were interested in it because it's been shown to target TGF-beta, TNF-alpha, PDGF, and collagen 1 and 3, which we know increase with age in the ovary. And so I will mention that this drug is used in idiopathic pulmonary fibrosis. It does have toxic side effects in human, and so it would not be something that we could translate directly into human, but again, this was important proof of concept. And so she first wanted to see whether or not this could have legs. And so the way she did this was treating primary ovarian stromal cells with TGF-beta, which we know induces a fibrotic response. And then she co-treated with pirfenidone to see if she could abrogate a fibrotic response. And so what you can see here is the levels of collagen expression. So with TGF-beta 1, these ovarian stromal cells increase collagen 1 expression, but co-treatment with TGF-beta or with pirfenidone is able to abrogate this response. So this tells us at least in vitro ovarian cells are responsive to this drug. She then developed an in vivo paradigm using a mini osmotic pump to deliver the drug throughout the animal. And so you can see here in this validation study with a non-toxic blue dye that when we implant these osmotic pumps, we're able to deliver a substance throughout the animal's body. And so she did this in this experimental design. She treated mice at 7 months of age, either with control or pirfenidone, and she treated them for 6 weeks. And the reason why it was 6 weeks is because this is the maximum amount of time our IACUC will allow these pumps to be in the mice. She then took mice at exactly at the end of treatment to analyze them, but then she also took a subset where she removed the pump and allowed them to extend out 3.5 months, at which she looked at a second time point. And so this was everything that she looked at, which I will not go through all of this data, but she looked at weight of the animals, she looked at bone mass density, and then she looked at estrus cyclicity. And at the time of harvesting, she was able to look at serum for hormones and cytokine levels, as well as the ovaries for transcriptomic analysis, as well as histology. And so what I can tell you is that unlike the human, which is toxic, we saw no evidence of liver function concerns or toxicity, and no systemic changes in the animals in terms of the cytokines that they were producing. So what I will focus on is what I think is most exciting to this audience, which is what's happening to the ovary and the reproductive function of these animals. So obviously with a low dose antifibrotic, our hope was that we would be able to change the collagen content. And what you can see is that on the top with control animals in this period, there's a significant increase in collagen as we would expect. But in our pirfenidone treated animals, there still is an increase in collagen, but it's not as dramatic. So this worked from that perspective. But what was really exciting is that when we look at the transcriptomic signature, this is 3.5 months after we've removed the pump and after drug exposure, what we see is that there's about 300 genes that are differentially expressed in these ovaries in animals that were treated with pirfenidone. And if we look at the pathways that are affected, what we see is that there's a downregulation of inflammatory pathways and an upregulations of pathways that we would like to see in terms of reproductive function, like, for example, cholesterol biosynthesis. So this tells us that a low dose treatment for a finite period of time can be stopped and still have lasting effects on the ovary in terms of ovarian aging markers. And what was really exciting was when we looked at ovarian architecture, we saw changes that matched with what was happening with the endocrine profiles. So at the top, you can see, again, control animals at 8.5 months. By 12 months, you have an ovary with a lot fewer follicles and a very fibrotic stroma. But in our pirfenidone treated animals, what you can see is that there's a maintenance of follicles. And again, drawing your attention to those large circular structures, solid structures of corpora lutea, you can see that we see more corpora lutea as evidence of more ovulatory events in those ovaries treated with pirfenidone. And this matches what we see with the endocrine profiles, where there are higher levels of AMH, which are produced by growing follicles. And we see a trend towards an increased production of progesterone. And then when we look at estrus cyclicity, we know with age, as you can see in the black bar, these mice with aging become acyclic, but with pirfenidone, we're able to maintain cyclicity of all of the animals. And so we think that this is extremely exciting, and we're currently looking at fertility in these animals, but also thinking through how can we translate this with more safer methods to the human setting. And what I will tell you is that there are drugs that have antifibrotic properties in the ovary and other tissues, such as metformin, which have a very high safety profile. And so we think that this could be a really important proof of concept drug to test in this scenario. So I think it's important as we try to move towards translation that not only can we modulate this parameter, but can we measure it? And so we are interested in this because it could be a biomarker for ovarian aging, but a way for us to also assess emerging therapeutics. And so we've been looking at this from the standpoint of how do we measure stiffness non-invasively. And there's a technology called shear wave elastography. And the concept here is that you propagate an ultrasound wave through a tissue. In a softer tissue, this wave will propagate more slowly, in a stiffer tissue more quickly. And this technology has been used in a clinical setting for various non-reproductive tissues shown in gray, as well as various reproductive tissues shown in blue. Now, this technology has been used in the ovary in the setting of polycystic ovarian syndrome, but has not been used in the setting of reproductive aging. And so we were able, through a grant through the Global Consortium for Reproductive Longevity and Equality, to purchase a device ultrasound machine that's able to perform the shear wave measurement. It is now in our fertility clinic here at Northwestern. And what you can see here is that we can use a transvaginal ultrasound to make these stiffness measurements. And so that's an ultrasound of an ovary. The black regions are follicles. These are fluid filled. So these are spaces that you would not want to make measurements on. But we can use high confidence maps to define regions of interest where we can trust those measurements in the ovarian parenchyma. So you can see those circles are regions of interest, and we can get corresponding measurements of stiffness of the human ovary. So we have a trial now that's ongoing. We've so far made measurements on 54 individuals. These are women, sort of all comers, who are coming in for infertility treatment or fertility preservation. We make these measurements on women at their diagnostic scan. So they're on the early follicular phase, so same stage of the cycle. This is prior to any hormone treatments, and we're excluding patients who've had IVF within the past year or have any pathologies known to impact ovarian stiffness. And our primary outcomes are measuring the stiffness to see how this changes with age, but we're also collecting a ton of secondary outcomes so we can correlate this parameter with reproductive outcomes. So I have been taking this data on the road. So you might have seen this before, and every time we update, so the significance kind of toggles. But I think what you can see here is that we're measuring various parameters of stiffness. So we look at coefficient of variation. So this is the size of the standard deviation in relationship to the mean. We also look at elastic heterogeneity, which is the difference between the average of the highest 3 measurements and the average of the 3 lowest measurements. And then we also look at the maximum value of stiffness from an individual out of about 17 measurements made per each participant. And so again, we're at around 50 participants, and we're always hovering around significance. But I think what you can see clearly here is that there is a positive correlation between age and the stiffness, these stiffness parameters of the ovary. And this is super duper preliminary. We've been doing this now for about a year. So you have to remember, these women are coming in for a diagnostic scan. We're then tracking them in terms of their reproductive outcomes. So going through IVF or IUI and ultimately getting to live birth. So it's going to take us a while to accumulate this data. But I think it's super interesting here that there is, again, three points, but there are 3 points, which shows that there is a negative correlation between parameters like fertilization and percent euploidy. So it will be really exciting if this continues to pan out as a potential marker. So I want to end with just a few minutes about how do we model this environment? How do we continue to drive basic science forward in this space as we're also trying to move forward clinically? And so I did spend a lot of my time in Teresa's lab learning how to grow follicles outside of the body. And this is a tremendously powerful technique. But I was also interested in figuring out how can we culture the other side of the equation, which is everything but the follicle. And we know that the ovarian stroma is highly complex with all of the different cell types that are listed here. And Teresa's group was really pioneering in being able to actually culture primary ovarian cells, stromal cells, but she was doing this in a 2-dimensional culture system, which was great, except over long periods of time in culture, you essentially are getting a mostly macrophage population. So there really was a need in the field to develop a system that will maintain cellular heterogeneity. And so through our T32, we have these academic accelerators where our students can go and do sabbaticals. And so one of my students went to Julie Kim's lab, and she was doing endometrial organoids, and she learned how to make organoids and brought that to the ovarian stroma. So the idea here is that she takes ovaries, and we enrich for the stromal fraction by poking out follicles and granulosa cells. We then digested the stromal enriched fraction, strained out undigested matrix and large follicles, and then we did a differential plating step. So oocytes will float, the somatic cells will adhere. We then lift those cells and we plate them and seed them into these microwell inserts. And you can see here, these are 96 beautiful ovarian somatic organoids. And we've done lots of work to show that various media support the growth of these organoids. But I think what's super exciting is that when we look at proliferation in these organoids, that they're happening really throughout the entire organoid. We don't have a necrotic core in this system, and all of our dead cells seem to be out at the periphery of the organoid. We also did single cell to see what cell types were actually in these organoids. And this was really one of the most exciting. I think I'm going to get this framed when this is published. But the fact is that we have a lot of ovarian somatic cells that we would expect to see, again, in our system. So all of the various cell types from endothelial, epithelial, immune cells, luteal cells, stromal mesenchyme, theca, and we do also have some granulosa cells. What's really exciting is that when we look close at what's actually proliferating, we were concerned that maybe it's just one population that's proliferating. What we see is that there's actually proliferating cells throughout all of the different compartments. So it says that we're really supporting the preservation of these ovarian cell types. And in fact, we see them both in the input monolayer, but also persisting through 6 days of culture. So these organoids self-organize, which I think is always a really interesting concept, but we clearly see in protein markers that we have preservation of these cell types. So we see fibroblasts with vimentin, we see F480 positive macrophages that are outside at the periphery where our dead cells are. We see granulosa cells throughout, and then we see steroidogenic cells that seem to be excluded from the core of the organoid. These organoids are functional, so they produce hormones that we can actually detect in the media over time, so estradiol and progesterone, and they also produce matrix components like collagen and hyaluronin. And I think this is going to be a really useful tool because we've been able to show that we can also make these with non-human primate tissue. This is with collaboration with Mary Zielinski, where you can see that we have organoids from both the cortex and medulla of the rhesus macaque. And again, with our various markers, we can see that these cell types are also preserved. So I think there's no reason to think that this can't move on into humans. And I'm really interested in using this as a potential complex phenotypic screening assay to move our work forward in fibrosis. So our organoids will respond to TGF-beta. They will induce the expression of fibrotic genes like collagen, smooth muscle actin, and periostin, so we can screen for compounds that block this response. We also know that with age, they don't aggregate as well, and this is extremely robust. You can see young organoids aggregate quite nicely, and old organoids do not. And so again, this is a screening parameter where we can look for compounds that are actually able to restore this aggregation potential. And finally, this is work that Riva, one of our newest master students, is going to take on, is what happens for long-term culture. So we try to move the needle forward from the Woodruff studies, from being able to sustain more than 6 days to not just have macrophages, but now we want to see what happens when we culture them. And again, what's really striking is that now they can grow for 21 days, and we're now just at the limit of what they're able to be grown in. So we think that there may be no limit to recreating that somatic environment. But what's really, truly remarkable is that when we look at dead cells, there's really no death in these structures. And what you can see is that these cells have now spread out. And in between, we have beautiful matrix that's being deposited. So again, this is a really useful tool to see if there's compounds that can block accumulation of matrix. So with that, I will conclude. I know I took you for a wild ride, but in the last 10 years, we've been able to show that ovarian inflammation, fibrosis, and stiffness increases with age. And I think that this is in large part due to failed debris clearance mechanisms, which again makes the ovary a really special model for studying aging, where we know debris clearance failure is a large part that's driving aging. Fibroinflammation has important implications for ovarian physiology, follicle growth, gamete quality, ovulation, but also potential implications for pathologies like ovarian cancer. We think ovarian fibrosis represents a therapeutic target, and we're actively looking at agents that can target this specifically for the ovary. And translational methods like shear wave are really important to make sure that we can evaluate the effectiveness of these therapeutics, but also as potentially a novel biomarker. And I think these ovarian somatic organoids will really help us accelerate identification of new antifibrotic compounds and pathways. And so I acknowledged everyone first, but I also want to again acknowledge this work with all of my lab past and present collaborators and the diverse sources of funding that have allowed us to explore. Thank you very much.
We have time for about 2 questions, so we'll take one from the audience and one from online.
We can also go.
What?
We can have an extended.
Oh, you can go over? Okay. So we'll start in the room. Teresa.
What a tour de force. That was just staggering and beautiful, and congratulations. And the next decade is going to be even more amazing. I'm very convinced of that. So my question, so the anti-fibrotic work is just elegant and very compelling. I wonder if you take the other approach, which is to block ovulation with GnRH analogs, so that you don't have ovulation. You can do that in the mouse fairly straightforward and see if you can delay all the signatures of aging into a later time frame. And the human equivalent for your shear wave measurements would be young women who take consecutive birth
control,
pills
yeah.
to block ovulation for multiple decades. And I wondered if in your 40-year-olds, which looked relatively young, if those had relatively fewer numbers of ovulation, or maybe had that past history of blocking menstrual cyclicity for some period of time during their reproductive lives. So anyway, just a brilliant set of studies.
Yes, and I think that's a really important question that we need to address and really dissect out. I still think that you're going to have aging happening because of atresia and the cell death that's going to happen that pathway, but you would certainly predict that if you didn't have a CL and you didn't have ovulation, that you would minimize the burden of some of these mechanisms. And then your question about contraceptive use is really interesting, and it's just difficult to control for with the data that we're able to collect, but that's not a reason that we shouldn't move in that direction. I've also been asked a lot about what like hormone replacement therapy would do as well. So I think looking at these hormonal interventions could be really interesting.
Amazing stuff. It's just so fascinating for me as a clinician to see the interplay with what we see and struggle with in these 40-year-old women every single day. Along the same line, if you have women who are on chronic OCPs, continuous OCPs their entire life, their age of menopause is not different or not tangibly different. And so suppressing ovulation, I think, is probably part of the equation, but the age of final menstrual period is pretty fixed. And so I don't know that we're going to see huge differences, but we could certainly study it.
Yeah.
Like asking women the cumulative dose of OCPs they've taken entire life and the age at which they have menopause.
Yeah, and I think that would be super interesting to do, because I think that's right. Like women who are in OCPs don't have a, delayed menopause. Yeah. But I think, again, it's because you have all these like I pulled out all these images because we've stained sort of cell death in the normal ovary, and I wanted to show that. I just didn't have time, but like there's just a lot of cell death. So I think it's a combination of multiple factors, and we didn't even get to the egg, that part of it. So yeah, but I think it'd be really cool to design more controlled or rigorous studies to capture this type of data. Question.
So like what the actually to ask in the other aspects of movement, she must run a lot of material. So now to get things young, the things young, protection is appropriate reproductive response. So are these mechanisms relevant in all or in a subset, and can we actually look into if there are clusters which they just show accelerated rates compared to other mechanisms?
Yeah, so I think it's going to be really interesting. We haven't done formal studies in POF like populations or genetic risks, but I think it'd be really interesting to look at that. I do know that, for example, like in PCOS or obesity, like there is fibrosis and inflammation that are happening in the ovary. Whether it's happening in these other conditions, who knows? And whether or not they're the same mechanisms that are driving it or different, we have to look. But that's a really good question.
I'm going to take questions online.
All right, so the first question from online. So in terms of the effects of ovarian stiffness and extracellular matrix on oocyte quality, have you looked at integrins in the follicles and or RGD motifs in the extracellular matrix associated with follicles, and how they might differ during ovarian aging, as these are the primary way the cells bind to and communicate with the extracellular matrix?
So I think this is a wonderful question. So how does the follicle actually integrate the signals and the cues that are happening from the matrix, and how does that ultimately translate to egg quality? And I'm going to defer you to, or invite you to speak to Dr. Farnaz Amargant, who again came from my lab, and she's studying that very question as part of her lab. And so I think she's probably in a much better position to answer that really important question.
And we have a question from John Eppig. So is fibrosis accelerated in ovaries of mutant mice with precocious reduction in the number of follicles? If so, does this mean that follicles suppress fibrosis?
Yeah, so that's a really great question. And this is from John Eppig, who is my idol, egg biologist. So John, I would say that I think that that's the case, and I'm largely basing this on work that we've done with Steve Ward, where he had a oocyte-specific knockout of XOC5 that actually caused depletion or loss of follicles. And what you see when you lose follicles is that you have an acceleration of this fibrotic response, which sort of begs the question of, are the follicles potentially signaling or keeping that stroma young? And so I think that's a really important possibility that cues from the egg are actually keeping that stroma in that young state. And I think our organoid model will actually help us address that, because we can now bring oocytes back into the picture and see if we prevent some of these age-related changes from happening.
And then one last question. So speaking of organoids, can these ovarian somatic organoids be cryopreserved? And do you know if when thawed that they still have functionality?
That is a really great question, and I don't have an answer for that. And I think that again, thinking, I don't know where this person is kind of going with this, but I would imagine that you're thinking that maybe this could actually be used sort of therapeutically in the setting of fertility preservation to save the stroma and then potentially co-culture. So I think maybe, doing those studies is going to be really important.
So the last questions are from trainees. If you have a question.
Yeah, I was just gonna ask, what role do you think follicular atresia plays in ovarian stiffening? Like, would you see increased stiffening because of the diminished debris clearing? Or would there be kind of a decreased stiffening because there's a lack of ovulation?
So I think when you have, I think we would have to actually test this. So the question is, like, with age, do you see more atretic follicles that are accumulating and not being cleared? And then my prediction would be that you would be recruiting immune cells to try to get rid of that debris, which would then cause inflammation, which then leads to fibrosis. So my prediction is that down the line, an increase in atretic follicles would lead to increased fibrosis, but it's like an indirect outcome.
One last question from trainee.
Congratulations, Francesca. I was interested in your populations of novel populations of cells that you're describing, like the Ovarian and the multinucleated macrophage giant cells. And I'm just wondering, like, are these seen in humans, and what's the extent to which you see these populations increasing in human nature?
Yeah, so we have seen the multinucleated giant cells in human. I actually think so. I think it's probably related to the length or corpora lutea, sort of the number of corpora lutea, but also how long they persist. And so I think we need to do a much more detailed characterization of the populations where we see these structures, and then what are the characteristics of ovulation in those particular species. But we do see these populations, non-human primate and human.
So I imagine there's a lot more questions, and Francesca would probably answer them. If you have a pressing question, send it to her. Otherwise, if you could help me thank and congratulate Francesca.
So thank you also everyone online. And I also think that this is recorded. So if it went really fast, you can always hear it again. But I also want to invite those of you who are in the room. We have a lunch reception afterwards, and we can continue these discussions. And thank you, everyone.